Of course. You know, yeah. No, it's good though. It can be. You know,
All right, let me welcome you to the 2024 season of our bioseminar series. You know the routine by now. You stop out there, sign your name, choose one of those delicious, if not homemade, at least made cookies, and then you come in here and enjoy the seminar. So, it's my pleasure to introduce today's seminar speaker, a 2013 grad of Carleton. And part of it, he was sitting in one of these very chairs here not so long ago. So he's done that. After leaving Northfield, Keenan went to the University of Nebraska Medical Center, where he got his PhD in cancer biology. After that, came back to Minnesota, to the Mayo Clinic, where he did his postdoc in the division of hematology and oncology. Since the postdoc, he's been a teaching faculty member at another MIAC school. All right, we'll say the name, Gustavus. But since then, quickly moved away from the MIAC, and in the fall of 2020, accepted a position as a professor at the Minnesota State University at Mankato. His research there has focused on using genomics to, maybe I'm summarizing this too much, but using genomics to enhance the effectiveness of personalized medicine. I'm going with that. Good enough?
We got all the buzzwords in. We're good.
Okay.
Yeah.
Keenan, I'll let you do.
All right. Thank you so much, Stefan. And yeah, it's super cool to be back. You guys are in a super special place. I won't lament too much. If y'all want to come out and do questions after, happy to share some fun Carleton stuff too. And like all the stuff we used to do, stuff like that. But let's do some sciencey stuff too. So fun, hilarious title designed by me, patent pending, hooray. But for the most part, I had the little Dolly Mini make me 3 little phases of what I try to do. I try to use genes to predict the future. I try to see what cells are affecting that future. And then ultimately we try to change the course of that future with drugs. I will admit, without a paying account, they're not that great, but that's okay. We'll make the most of things. But my main goal again is sort of to make that more perfect union between scientists and medicine to connect for that patient and hopefully guide us toward a future where each patient is treated differently in a way that can most effectively treat their specifics. So hopefully everybody's obviously known immunology. I tried to make this an immunology seminar, but I toned down myself. But what we're going to take a look at today, there are cells in your body, B cells. Ultimately, what they're going to do, their main goal is to make one of these giant antibodies right here that will go and bind to whatever enemy is threatening yourself, your body. Now, the journey to that naive B cell to the adult plasma cell that's spitting out those antibodies, they go and create these. They're going to go find all the issues, things like that. They're going to bind all that bad stuff. It's a tumultuous journey. It's involved with a lot of stuff. There's certain things that happen in this development of this very lethal weapon that you have that doesn't happen anywhere else in your body. And again, as a B cell person, I'm going to really scoot past all the fun stuff. And I'm sorry, come find me some other time. We're going to talk all B cells. I'll convince you they're way better than T cells. But again, this is not on the exam, but there's some things in here that do not happen elsewhere in your body. To make the best possible antibody, you're going to mutate genes to make them that much stickier. You're going to be shifting and moving whole classes of genes around, chopping DNA, all kinds of stuff. That doesn't happen anywhere else in your body. So this is a very dangerous, crazy cycle, because the whole point is sort of this forced evolution, that if this new offender enemy, we're going to have to basically see how our pathogen works, how it binds. We're going to switch some things around. We're going to do some cycles over and over until we get the very best number one. Y'all ever hear this? It's the example of organisms. It's a shark where, like, I think in the womb only one fights and survives out. It's kind of like this. So this is aggressive, and there's lots of stages to these things, and you can sort of genetically age a B cell from the left to the right as well. I don't get to do all the fun memory and plasma stuff today. We're kind of going to be seeing in the middle where those red guys are. But here's where all the sort of summary shows up. Oops, mouse. So the drive is to figure out who exactly in aim number one is going to have a bad time. If we can identify those patients, we can take a look and see what are the actual issues driving those bad patterns that we saw in the stats and the genes. And then my favorite part, and my students' favorite part, is we are going to try and propose, based on the genes that are the offenders, a new route, a new drug combination. There are so many active drugs right now that there's so many different combinations we could do. I would never be able to get to all of them. So in essence, I want to find and use info. I want to validate that inside a cell or an organism. And then when the time comes, I want to reverse that course. I want to rescue that. Now, I'm not at a hospital. I don't have patient samples, but we can still do, in the age of big data and sharing, we can make a lot of moves here. And that's kind of the goal of some of this work. Now, to begin again, not only is this not an immunology class, it's not a cancer biology class, but here we are. Wanted to showcase some of the existing hallmarks of cancer right here. The two biggest ones as a cancer biologist that I always look towards is you have got to survive. Where is that? There it is. You've got to grow. Those two are the main ones. Now, have you ever heard of the story of the mice and the elephant? All blind mice is like, no, it's this, and they're at different parts of the elephant. Same thing with cancer research. There's plenty of people that say it's a genomics disease. No, it's an energy disease. No, it's an immunology disease. Here's the problem, it's everything. That's the problem. And we'll get into treatments, cures, all kinds of cool stuff. And at the very end, I want to talk about some really cool frontiers we're at. But for the most part, we are also going to interact with this one today a little bit, the immune cells. So basics, hooray. If you get a mutation in DNA, RNA, protein could be different, right? If I mutate that gene, it could be so odd that it doesn't form anything at all. Even worse, or sometimes just as bad in some cases, it could be abnormal. It might bind the wrong thing. It may never stop functioning, all kinds of fun stuff. So these gene changes, they sort of inform how a tumor behaves. And not every tumor is going to have the same combination of these. Eventually, I remember this from genetics in here. No, it's okay. We were actually late. I don't know why. It's okay. It was a great time. But one rare mistake, because your cells are pretty good at not making these mistakes. One mistake may just cause that cell to go a little faster in mitosis. It's not harming anything yet, but it will outcompete all its cousins. And suddenly, in that big pool of new cells that are just that much faster, maybe a second mistake. And that mistake allows it to survive a little better. Down the line, over and over again, the advantage grows and grows. And eventually, we end up with the bad stuff. Now, this, unfortunately, is where we usually see patients. The symptoms are showing. That's too late in a lot of cases. Our work doesn't detail cancer tumor initiating cells, or the more popular way to call them cancer stem cells. But a lot of some of this genomic stuff, looking back in the past, part of the journey that we'll see, maybe not today, but in the future, in the next 10 years, we'll be looking and seeing predictions way in the past with this. But time to meet my main adversary, diffuse large B-cell lymphoma. So your B cells can go bad. I showed you that process. They're under a lot of pressure to evolve the very best B cell. That means chopping DNA, all kinds of stuff. Our stages and our direction in this disease is very different than normal. So normally, if you have a stage 1 to 3 tumor in any of your other organs, right? Let's say pancreas, lungs, one of those. Surgery is an option, right? Get it out. Not a stage 4 because it's metastasizing. Here's the problem with ours. They're already everywhere. There's no surgery option here, and surgery options are extremely limited. So our field has always had it. That's why hematology is always kind of on its own boat with oncology. We always have to have a completely systemic approach of some kind. Now that's comes with some good things, comes with some bad things. But to say the least, it makes it the zone that a lot of crazy experimental stuff can be used. And we'll kind of get to meet one of those today. Now, the initial, this is from 2001. Looks pretty fancy for 2001, and it was. We took these genes in these patients, and we wanted to say, can genes tell us who's going to fail and not? They, again, not on the exam. But we found one profile that had a gene expression of certain genes up on the top with red. They were more aggressive. So when I showed you that big like recycling development stage, these are sort of the adult ones, those ABCs. They're the ones that are out to get you. They're the ones that behave like a typical cancer, and they're going to chew up and parasitize everything. The GCBs, they're still cancer, but they're strange. They're just sort of stuck in that cycle forever. That can grow into a lethal tumor, but for the most part, we did find for the first time the genomics could somewhat predict course. Using a lot of this, we were able to put together regimens that could target these genes in here. We actually came up with something called R-CHOP. The CHOP is a chemotherapy regimen. Chemo's not the evil thing that sometimes it can get characterized as. It can actually do a really good job. Now the R over here, though, was an antibody, a synthetic one, called rituximab. This binds something called CD20 on the B cells, causes them to get killed by the rest of the immune system. Awesome. Now again, the other cool thing that's happening here, your immune cells are fighting your own cancerous immune cells, so a lot of wires are getting crossed at this time. But for the most part, you're looking at what our field looks at. This is if you get a diagnosis right here, and down here are the months. I mean, across, what are we looking at? Where am I at? We're at 48 months right here. It's usually a pretty good metric. Got about 65%, everybody's still alive. That's actually not bad. And if you look down here, that's 10 years. Not bad. So I do often have students that say, well, cancers can't get cured. No, they totally do. We totally have tissues that can get cured. This is one of them. So the field is off to a great success. Like we have, if you diffuse large B-cell lymphoma, you are looking pretty good from the outset. This treatment is good. But we obviously want to make progress. Now, as genes became more accessible and we could see specific mutations that would drive certain pathways, like this one right here, CD79B, if you get a mutation here, this gene is always on. It's always firing and signaling and saying, grow, grow, survive, survive, don't die. It's mimicking the signals that we saw in that development pathway that are usually on for the right reasons. Because every oncogene usually serves a pretty neat, there's usually important purpose normally. And eventually, with all these on, you're going to end up with one of these big transcription factor families like NF-kappa B, and that thing cannot die then. It's like a hammer, basically. So we eventually, with targeted sequencing, we were able to find drugs like ibrutinib, penzolobremenab, sorry, that could bind to specific mutations and stop just that gene. How great does that sound? Just your tumor has CD79B alterations. We're going to block it with a single drug. It's not going to hurt your other B cells. It's not going to hurt your other cells. The chemo will get the rest of them, and rituximab will get all your B cells. These are people, for example, when we talk about vaccine, people treated on this scale, they can't have the vaccine. Their B cells are getting erased right now. So I wanted to introduce the good news. That's at treatment outset. That's if you show up, that's your odds right there. Now, see all that space and all these drops right here, right? Let's look at those patients as a group. That is the drop. It's from 2017, 500 patients. At a year, look at about 30% people left. So if you fail that first round, you're in a bad way. That's not good. And that keeps dropping all the way right here. I mean, we're looking at 3 years right there, down to 20%. So the treatments at this stage, they're an area of need. And like I told you, we don't have surgery. We have to think big and weird and experimentally, drugs thrown at stuff, things like that. Who has ever heard of synthetic or living drugs, chimeric antigen cell receptor T cell? Something strange was turned to for these patients. What happens is a patient will show up with at this stage, R-CHOP resistant or refractory relapsed lymphoma. What an experiment team did at Penn, long time ago, took T cells out of the patient. In the lab, genetically modified them to express a receptor that would kill and target the tumor. We basically fanged and armored a T cell, and we put it back in the patient, and we saw amazing results. We unleashed a genetically modified hunter, basically, in an ecology system that wasn't ready for it. And these tumors were getting erased. That's the new curve. You guys, I mean, we'll see in the field, if you bump a survival curve up 2 to 3%, that's a spoken discussion at the big ASH meeting in December. But if you bump a survival curve up, what are we looking at? 60%? Where did we end? 30% the last one? Massive. So the field's excited. The field's so excited about CAR T. We have clinicians right now banking and saying we should actually, from the outset, do this. That's a little aggressive, though. Let me tell you a couple things that happened with CAR T here. This poor little patient right here, there's a certain cytokine storm. There are sort of signals from your immune cells that go aggressive. These are aggressive T cells that we've supercharged to kill your tumor. They're going to get they get a little out of control sometimes. Cytokines affect your brain, delirium, all that stuff. And that's not the only cytotoxicity that's associated with this. Or sorry, that's not the only toxicity associated with this. Who's ever heard of financial toxicity? That costs about $500,000 right there. And in the early stages in 2018, 2019, we're still all on trials. Most of that's taken care of in that sense, but now we're here. So there's a reason why we still want to find details in the genes. Now, one other thing that's been happening, and I know I've all crunched all these studies down. What we're looking at is that we started taking tumors and we had so many of them and so many sequences, started to be able to classify them. These are the patients up here. We started to see that certain gene groups, so I'll say gene, so up there, started to see that certain clusters of patients started to emerge and that they had the same sort of evolutionary profile, same features, same idea driven by the same genes. These studies kept replicating each other. They all kind of competed and fought each other. And eventually we ended up with like 3 names for each one. It's really confusing. You kind of have to share your alliance depending on which way you like call it. Anyway, I'm kind of an easy B in the red person right there. But the whole idea was here, and this is sort of at the same onset as the CAR T revolution is happening, is to say these genetic clusters should be treated with specific medications. And that's kind of showing here from the right paper right there, one of their classifications. Now, the cool thing that this also did, even though it was meant to inform clinicians, we found the responses were still very good when you targeted drugs that were very specific to those genes. We learned more just about the tumors in general and the evolutionary paths that we can take. Another key thing that we learned, and that's where I sort of came in at Mayo Clinic. This was my goal. And right now it still is because it's that big. The toxicity we found with CAR T. Now, before I say that CAR T's, there's anything bad, you all saw that 30% jump. It's amazing. Those are families, those are Christmases, those are everything. But would you rather know that you're going into a CAR T with less reliability? You would not want to enter that trial or that treatment if you knew that the genes you had in your tumor were not going to respond regardless. That's the goal. It sounds a little grim, but it's something that then we can say we can translate a better therapy right there. And clinicians can pick that up and target that for patients. This is not on the table anymore. So there's my original napkin. Yeah, chicken scratch. My writing has never improved. Thank you all, my old professors, for reading through this. Because now as a prof, I sometimes come across, and I put sticky notes on everybody's name, sometimes come across that writing, it's just like, oh no, I gotta get through this. It's okay, it's okay. So the key thing too, we can use regular patients to find this. Who arrives already at diagnosis, a profile it's not going to be CAR-T eligible. It's also probably going to fail at first line because those are the people we want to be able to target and see. So moving on from chicken scratch, a lot of the field, and this is that's why some of this is so quick and small and emerging, started finding patterns in a few very prominent genes. Now the thing is, most of the original studies were done with thousands of patients, but we're only working with people here that are past that first regimen. So you're only working about 40% of the original population, so it's a little smaller. I found a couple things here. Mainly, we're gonna talk about these two, because otherwise, I was gonna do some metabolism stuff, but I think we're all a little scared of that regardless, so we're not gonna do that. No Krebs cycle. Sorry. It is important, guys. So, now the key thing though, was one thing that it makes intuitive sense. CAR-Ts, they're supercharged T cells, but they're not just fighters and killers. They're the commanders, they're the C4s. They evolved into the entire army. We found that mostly, if you try to inject these into patients that had a very depleted environment, the immune cells are already defeated. There's no sense in sending in more soldiers. It was already gone. And these two factors that I'm going to try and point out here and how they did that. That was one of our goals. Because eventually, yeah, you don't want to end up here because that's just going to be tough, regardless with the toxicities we talked about. So what we're going to do, like I said, I don't have any patients anymore. I can't run RNA-seq on everything I see. It's a sad day. I remember at Mayo, everybody was like, we're going to run our RNA-seq on these 100 samples. I was like, neat, that's good. And now we got to rely on the data, the mining that we can do out in the, out in the supplementary data files that exist. But it is still rich, and it is still something that we can all put together. It's kind of neat. Now, here's the first one. So on the left, the survival curve of that original population we talked about. In red, if you mutate this gene called TP53, I'm just going to call it P53 so I don't stumble on it. We call it the angel gene for a reason. It is a very giant checkpoint for your cells to do mitosis. Should you lose that, your cells are uninhibited. They can kind of do whatever they want. So it's a tumor suppressor. So if you mutate that, you're in that red curve from outset. And then on the right, the pie graph, because like I said, the data is still pretty limited. Those are CAR-T patients. In red, do not respond. In purple, you're fine. So on the wild type right there, things are looking all right. A little over half the chance, like we saw, about 60%. Not the case if you have a P53 mutation. So why? So key thing, dug into some data, combined some sources, normalized them. I wanted to hunt for the right thing. Two things that I started with down here. Wanted to see EFS 24 means event-free survival in 24 months. I wanted to find out who survives 24 months and who doesn't, what genes go towards against survival, which genes are for survival. And then down here, I wanted to see what genes in the patient group that has P53 versus non, what genes are high, which genes are low. We get a nice little XY here. Each gene has a value for each one. Something showed up way in the corner up here. BRD4. Or as my wife likes to joke, bird4. It's proper, don't say that. But BRD4 is a transcription factor. It drives a lot of growth and craziness and survival. So it's huge. It's tons of it. There's way too many of it in the P53 people. There's way too many of it in the failure people. So I found my little target right there. If you just isolate BRD4 in general, let's say we made a population just one standard deviation away. Let's call them high ones. As you can tell, not looking good. Now the fun, colorful stuff up here. These are all the pathways, and I can't zoom far enough, sorry. These are all the pathways that are lost or gained when you lose that P53 as well. So we're gaining this giant oncogene, this BRD4. You are losing and you are gaining key cell cycle stuff, proliferation stuff. That was meant to be an arrow. Proliferation, all kinds of bad stuff. Tons of metabolism, we're gonna avoid that. You lose a ton of cell death pathways. They don't work anymore. You can't kill these cells anymore. And immune regulation, that's the big one in pink. And that's the weird one. If you lose P53, your immune system is not as good as it once was, or at least patients don't have the good firing one. So, fun times. I took both and I said, what genes? How do genes associate when you have high BRD4? And I'm also going to use the same metric, your altered P53. You notice two that were really important in a previous analysis. CDKN1A is a typical tumor suppressor. It's not fun to lose. It's bad. If you lose it, your cells are uncontrolled. They're going to grow. They're going to get you. This one was unique. TNFRSF10B. Say that 10 times. I'm just going to probably call it TNF from now on for a bit. TNFB. What we saw was this death receptor. It's on the surface of your cells. Your T cells need that to kill. Should it be lost, they're aiming at nothing. So what Durva and his colleagues found out was that of the most important genes for CAR Ts to need before they show up and they actually can make that kill, they need that surface receptor sitting on the surface of these cells. So if this gene is low, like we're seeing right here, CAR T cell just runs off. You can't kill that one. But if it has an attachment site, these, perfect, we're going to get it. And that connection with P53 might have been the key that we were looking for to say, what exactly is driving those losses? Now, another factor of this, did one more little analysis. We took both TNSFRB, sorry, let me zoom in for you. Took a couple of immune signatures. We did the right thing, kind of boring, but we said you have P53 and these genes for IFN up and stat up. That's just showing how powerful is your immune system right now. And you see in red, super duper reduced, down in the dumps, not ideal in both cases. Stout's the name of the group that did that. The next one, yeah, I probably need a new color scheme. Sorry, but I really like it. So, next cool thing we did is actually kind of a cool tech that is very accessible. So I have RNA data. But when you take an RNA, when you get an RNA sample, you aspirate from the patient, it's not just going to come up with just tumor cells. It's going to come up with some of your T cells, some of your macrophages, some of your dendritic cells. Deconvolution is a program that can tell me and give me a signature for each one of those. I can put that to each patient. So it's sort of a way to infer what cells are still running around the tumor. And this is that perfect idea. Like I said, you need to know what the immune system's up to. So I saw one thing happen here. If you're low death receptor, like that, TNSFB, and if you're low or altered P53, look who you're losing. You're losing your T cells, gamma deltas, little innate fighters, but you're losing your big CD8 killer cells. Those are your big key tumor ones. So each time, patients with that profile, they're showing up without the T cells. And without the T cells, you can plug as many CAR Ts as you want in there. They're not going to be able to inspire enough to fight. Equally, like I said, I wanted to see from outset who's going to enter that pool of really bad patients. If you have a P53 alteration, but you have at least some T cell signature. On the left, you're in black. Not doing too bad, but it's the ones that showed up with no T cells depleted. They're the ones that caved. Same thing with TNSFB 10-1 or 10B. So, same thing. We started to see a profile emerge between some of these genes that we've been talking about. That BRD4 oncogene and these two tumor suppressors on the bottom are TNSF death receptor and the typical tumor suppressor CDKN1A. You sort of see that nice inversion along each other, that in relationship. So not only are these two all connected with P53 down here, sort of this worst case scenario that these four things, BRD4 is being gained, the other three are being lost. You're losing all your T cells. If you put in CAR Ts, nothing's going to happen. And you're going to spend that money and you're going to have toxicity. So here's one thing in the drug world. Do you think it's easier what do you think is better to happen? Have an oncogene show up in high levels or lose one of your two copies of a tumor suppressor gene? You don't have to answer, just think. I don't want to put the pressure on. Because what do we have? We have unlimited drugs, right? If you have 8 copies of that BRD4, we'll see in a bit. We have tons of drugs we can throw at that thing. So any oncogene that shows up, we've gotten very good at making chemicals, drug it. But you cannot, it's very difficult to bring things back. Something, we had a plan. I saw this. One of my students made this fun little thing. It was not in PowerPoint. He wouldn't tell me what to use. It was something else. P53 is regulated or like sort of destroyed by a gene called MDM2. I'd ask Nutland, that I right there, it can destroy P53. So if any P53 is left, we'll get it back. Now that drug alone never had a good profile. A very cool chemical showed up. It prenopopped. So I just left it as an E. It is an agonist of mutated P53. It's strange. It's a very unique class of drug. We don't have many drug classes that bring back function of a mutated gene. That's its profile. So I took some cell lines, which are basically in a dish, in vitro cell lines from a patient in the past that can be immortalized. We could test them, do all kinds of drugs. This is much better than mice, obviously far better than humans. So here's our drug responses. As you can tell with each of these, let me go. In black, we have the EDS Nutland, we have Prenopop in white. And you can tell, I mean, as single agents, they will kill the cells eventually. They're not like one of those straight drop drugs that you see. But using Western blot to see the protein, we brought something back though. So whatever P53 was left over, or any non-mutated versions, these drugs brought it back enough to inspire the death receptors to come back, and they should be able to get killed by the T cells, which is strange here. This isn't actually killing our cells, but it is setting them up to be super duper killed by the CAR T cells, right? So we're in a very new age with medicine here, where we're trying to say, we want to set you up for success for the real thing. And we're going to start using drugs to prime patients, not treat them in some cases. So based on, we're also like, BRD4, clinking around in there, like causing trouble. We do have a drug for that. And we do have a drug to rectify the CDKN1A loss. I wanted to try a couple super aggressive drugs. Who's ever heard of a PROTAC before? We're going to introduce, it's very fun. Think of it for now, I'm going to show you a profile in a bit, but I'll think of it for now as just something that destroys BRD4. We also had, and yeah, sorry, it's still the science name, so it's this big nasty thing. Abemaciclib, that's like the second sciencey name for this drug inhibitor right here. So your CDKs are just mitosis promoters. They show up when CDKN1A, one of those tumor suppressors, is gone. But we really, really wanted to get rid of that BRD4. So BRD4 getting rid of is in red. Notice something strange about these curves. They're not really curves. Did you notice even at a touch with some of these, because on the left is the least amount, they're immediately dead in some of these. And the range of treatment is pretty wide. You are looking at standard deviation, I'm being honest, no standard error from me. But it was a very good single agent drug. We ended up putting these together. And this is where my students always love things. All right, when you put it together in a big quad plate and do combinations of each, this is where you generate synergy profiles. The red shows an area where, as a single agent, maybe BRD4 inhibitor killed 5%, maybe abemaciclib killed 10%, but in the middle, 70% died. That's synergy. That's what you at least want to see if you want to take something to the next step for patients, translation, but it does also show you partnerships in biology. It doesn't always have to be about treating. But yeah, we're pretty happy with this. It's always a big success because when you get the results, you always want to see it's just like, yes, we did it. And obviously, seeing is believing. DMSO is just sort of a nothing. There's abemaciclib on its own, still a big pile of cells right there. ARV alone, but at the same combinations, gone. Very nice. So this was presented and published. We're super excited about it. Hooray. But I did promise you a little profile, a little quick learning moment for the PROTACs. PROTAC stands for proteolysis targeting chimera. Most drugs you imagine, right? They bind to a target, just like kind of destroy it, right? A one-for-one match. PROTACs rely on something a little more sinister. Take a look at the diagram there. The PROTAC is still an organic molecule, but all our cells have a recycle system for genes that are no longer necessary. Ligase, ubiquitin, sends them down the proteasome recycler. The PROTAC facilitates that. It brings the oncogene, binds it. It has a second binding section that brings the ligase together, tags it with ligase, down the drain. It erases the gene, doesn't just bind it. Now, remember those curves that weren't curves and they just flatlined? The PROTAC recycles. It's not like a normal drug that just goes away after it's used. This means that, unfortunately, PROTACs are still sort of in early phase. They're very hard to control because once it's in the patient, it's going to keep going and keep erasing. Very hard to control that. And, whoops. So that's why, again, you would only use this treatment combination if somebody is devoid of T cells already, and we just need something. And that still is a chunk of that otherwise death curve. We're excited for that project, but at the same time, always have to tell the caveats. Part of science, right? So, project number 2. Oh yeah, I still got way ahead. This is good. Don't worry, I promise they're early. We'll be all right. It's been a long week. It's been a long week. They started you all early this year, right after New Year's. Okay, so number 2, a little less, a little smaller. Don't worry. MYC. MYC is a popular oncogene. Everybody's probably heard of MYC or seen it in the slides. It's gross. It's transcription factor. It's going to cause all the programs. We're looking to see the same thing, but from a different profile, different lens. Sorry that I use the colors again. They really smash. So I really liked them. Same thing though. MYC's bad from the outset. If you got high MYC, you're in trouble. Okay, or if you have the alteration. Looking at the CAR T patients again, you are in red, failing 2/3 of the time if you have MYC. Versus, and notice these numbers grew because this project's more in the future. I found more data sets to keep pulling in. And again, just the basics. If MYC is there, remember the purple, all the immune genes, we're going down. Everything's depleting, everything's turning into a desert again. Because the tumor thrives in that desert, parasitizes everything else with no predators around it to kill. Equally, there's been some really cool research about a little partner gene that's super annoying for us to treat MYC. MYC is already a transcription factor. It's almost impossible to drug specifically because it doesn't have that pure active site. What we can drug, though, is this goofy little thing, XPO1, exports in one. So when you make something in the nucleus out of RNA, it has to get pushed out to become protein. We can't necessarily drug MYC, but we can definitely drug that. And a lot of biology is showing that a lot of the responsibilities of MYC can be stopped right at this moment. So we had that in our head, especially as that we saw, again, when you have high MYC, each one of these in red is one of those immune genes that in the high MYC population, they're going down. Straight up CD8 signal, not good. That red population, a lot lower. Each time with one of these little corrected multiple hypothesis t-test, again, CD28, another T cell signal, ILR7, or another T cell signal, cytokine CCL2, and CD8's actually NKs, but it does go to show how devoid we're looking, and then just straight up stat activation. So for the immune system. So all those red ones showing the same thing. One other set of data showed up in between these projects. Schaller 2022 was the first publicly available. We treated people with CAR Ts, and we saw the ups and downs of the genes. So this is the first time this data has been available. So we can see what goes down in responders, not just people from the outset. So I wanted to check here. This is de novo association, which means how does that gene behave at that first diagnosis and who fails on the bottom. So I should, put this. So these are going to be high in failures or lost in this case. And who's getting lost on this side on that x-axis too. So if we look and sort of zoom at these graphs, and if we notice, I also classified patients based on if they had high MYC or if they had a complete absence of CD8 signal. From the outset, there is a giant pool of patients, and you can see some repeated genes in here. There's a pool of patients that show up with a complete devoid immune system. It's already bad. It's already depleted. It's already cold, as we put it. But a lot of these genes, some of them we saw up there, 7, CD828, CTLA4, they all shouldn't be lost. And when they get lost, the immune system is basically saying it doesn't have any defenses anymore. So if you send in that CAR T, we're going to be in trouble. Now, partnering up again, same analysis, sorry, but like I said, I speak to clinicians, and X and Y plots are very good ways to speak to clinicians, and color is always fun. Happens again, though. If I classify patients on that little partner XB01 that's helping MYC, and I classify them on MYC, we again see that CD8 T cells are the ones that are suffering, both in this case, between that partnership. We see a pretty decently significant relationship between gamma deltas. And strangely, we see a loss. It's not strange that we see a loss of those M0 macrophages. They can be good guys. I will admit, as an immune person, it's strange to see the neutrophils get lost. More on that later, I suppose. But again, should you be a patient in this second profile that we were describing, if you have MYC, but you do have T cells, or if you have a MYC alteration, but you at least have the T cells functional running around, you're okay. It's when you lose both at the same time, then you end up in that red curve. Significance maintains if you combine all three of those curves above the red curve, or if you just do mix, etc. So again, the field, my field, and it's sort of not sad, but it's how science moves. My field's moving further from genomics of this inside of the tumor, more towards what's outside the tumor. Who's calling the shots outside? Who's letting this thing grow? And here's the other analysis that we could then do with that new data. I took the people that were surviving for 24 months, and I redid the analysis. And I paired that analysis up against the people who died or had an event before 24 months, the low responders. The CD8s are still bad in both populations. But again, the ones that call the shots, that purple right there is the signature that's pulling from the really bad failed patients. So yeah, you do have to lose your CD8s. But if you lose your like command generals, the big system centers, the CD4s, then it's really bad. Then everything's gone. The communication lines have all broken down. Ooh, who's seen that? Sorry, time for a reference. Who's seen that Netflix movie, Leave the World Behind? It just came out. Yeah, remember when all the communication went down? Yeah, it was creepy. It was unsettling. That's like if you lose your CD4s. All right, it's my only movie reference for the day. So, like I said, this is super colorful and awesome. Two groups have published something that's now focusing not on the intracellular genomics of the tumor, but everything around it. We're calling these ecotypes. And the patients don't just show up with a tumor profile with genes, everything around it is what we can measure now. What we're seeing here too, the state and the activity level across these types, it's going to depend on macrophages, dendritic cells, T helper cells, and where those T helpers are. Each factor is sort of going to inform, as both of these groups sort of want, how to treat that specific group. Seems like I'm just adding a lot on the plate, right? We're just going to have to sequence like and do everything for every patient at this point. But yeah, I'll be the first to admit, these are just basically XY plots, but they look really pretty. And I was enjoying messing with these. So like we did with XB01 and MYC, we can see these different cell states up here. Now that this data is out, I could assign a cell state to each patient now. And I also put EFS 21 or EFS 24 fail. You're a larger bubble if you fail treatment. So even though these are mainly just for visuals, you see that big red amount right there in those high MYC and high XB01s? That cell state right there, number one. The bad guy right there. Again, let's take a look at CD8. That's your T cells. See some of these big red ones right here dropping like that? Same thing. Same with the ILR7 as well. And using these new data, I'm able to see that those key T cell signatures are in that S1 ecotype, and this does agree with their results as well. And that's going to sort of match up with that MYC group that we're talking about here. Again, seeing that that S1 is one of the more powerful MYC groups right there. But this is definitely still sort of a new frontier for us. But no matter what, MYC and XBO1 keep showing up. I'll highlight them down here, it's big. MYC and XBO1 keep showing up in what we're starting to call this cold or depleted patient. It's a patient that's showing up with tumor, and there are no T cells, macrophages to speak of. It's all gone, it's all tumor. So, of course, we wanted to drug things, so let's do that. So we have high MYC. We want to propose a treatment. We wanted to get it from both ends. We cannot drug MYC itself. Doesn't have the sites. It's too wrong, it's too awful. But we can kill it before it gets out of the nucleus. And at the top, we can get rid of one of its big products, CDK9. So we decided, let's throw these two together. And trust me, even though I'm showing you the good synergies, there's been plenty of failed times. So don't think that every time it's like, we got another synergy. It's a big event. But you can tell here, these drugs are a little more next gen. You will hear the word selinexor and AZD running around, AstraZeneca for this CDK9 drug. The selinexor is a very popular XBO1 inhibitor, and people are very excited. So we were excited to see some synergy with this. You see some of these red curves right here with the AZD and selinexor inhibitor, completely down. When I started my PhD, those max levels on the right were where I would always sort of like have to approach, like my macromolar amounts. We're starting to get drugs that kill at nanomolar. It's cool progress to see. That's like one of the best parts of science, seeing this stuff like move so fast. So again, like I said, this is ours, this is mistakes, this is everything else. But again, we found that the system worked. We found information. We validated what was going on in there. And if you do show up with MYC and no T cells, would this be a combo that could work? Yeah, definitely. So that's always the cool part, is being able to assign and see maybe someday in the future, even if this doesn't treat or do clinic stuff, still helps learn about the biology and the partnerships. And that's kind of the cool part with science. So, hooray, we did both of these. I'm excited to maybe do some more metabolism stuff. Guess what's really hard to drug though? Glycolysis, oxidative phosphorylation, because guess what? we're all using it in every other cell. So we're working on that. We'll see about that someday. But essentially, if we can identify right here, we can find a different route. And that's what's meant by the buzzword precision hematology oncology. We want to be able to know before we set you up to take CAR T that we have another route we could possibly take instead of going down this road. Now, the important thing, especially anybody that does stats, but any clinic stuff, stats predict populations, not individuals, but they're there for a reason. Hooray, though. Now, my meeting in December at the annual ASH meeting. I love going because I learned so many fun things. Here's a cool one. So so we're dooring. Like now's all the like relaxing, fun, cool stuff. The data's done. It's okay. No more colors. So macrophages are absolute villains. We found out they are the ones pulling all these strings in the cold phenotypes. They're totally horrible. They vilify and like zombify the other macrophages into promoting the tumor. They start treating it like itself and to defend it. That's bad. There are drugs that we have thrown on the shelf because they didn't kill the tumors well enough. We're finding that the drugs, especially if they affect the epigenome, can sort of turn those M2 bad macrophages back in time and sort of defang them. Very cool. Next, the CAR Ts themselves. We're starting to find drugs that can sort of make them more aggressive or make them more effective. Like we saw with ours. If we can inspire that death receptor back on, why not add it? It's going to make the CAR Ts that much better. But here's my favorite. It's the last one. It was a very cool thing. So I don't have the capability to do this data. All the RNA-seq that we have is bulk. But we have a new tech called single-cell RNA-seq, where each cell passes through is RNA-seqed itself 10,000 times. And you can see this amazing picture and portrait of the tumor microenvironment. The people that are doing the best on CAR T have this strange population of B cells, about 5%. Now the tumor's B cells though. So they were like, what's this? It was a healthy set of B cells that were surviving the tumor. It was somehow still fighting back. They were priming the T cells to keep fighting, and those patients were having success. It was like an insurgent population. It was like a resistance kind of. I thought it was so neat because at first when they saw that little 3% of B cells, but everybody that was doing well had them, they're like, what is this? But it's your healthy cells still trying to fight back, still trying to do the right thing. So that's a frontier that we're very excited about. Equally, hooray! Also, dog pictures. Wonderful. And the other cool thing, again, thank you all so much. And it's such a team effort. Like I have so many great minions that deal with me and my jokes and all that. Hooray. Great, great support from the department, everything like that. I also do bioed, it's fun. And notice there's something on one of the last things right there. I don't know. Adam wasn't actually a biology major. He's a computer science major. So now I'm going to really kick this bioinformatics. I'm going to start using that word for real now. It's going to be good. Anyway, but it's just another example of like the Carleton kind of family that we have here. And I'm so happy that you guys joined me for this. Thank you.
Have any questions?
Yep.
We got
Yeah.
time.
So it's important one. Seems like something that would be really tricky to know, because it also replaces.
Oh, yeah.
So how's that work? Where's the substitute coming from?
Oh, it's not. We're taking down all the good guys too that it's helping.
Uh.
Yeah, that's the best part of biology, like small biology medicine. Like, remember that BRD4 thing that I drugged that was really bad overall? That still is also helping some other good genes as a histone factor. So kind of get it under there. Now, as we play with the epigenome, or because I was going to do right here, I was going to do kind of a Venn diagram. I was going to say bad genes versus good genes that we're taking down. Right now, the equation is better for us to get rid of XPO1 and its partnership genes. But like anything, we're going to then create weaknesses by using that drug. And we're going to turn that population into an export and treated population that may now have a whole new profile now. Yeah, definitely one of the consequences of messing around with these.
Could you talk a bit more about those, like that population of B cells that resist the cancer? Like, do we know like what kind of cells they are or what they have and resist them?
Yeah, let's go back here. So in this development phase, there's certain interactions that happen with the B cells that actually allow there are T cells in this interaction. I was about to highlight yellow and yellow. Oof. With the healthy cells that are still doing this normal, they are still giving them the signal to those T cells and the soldiers that they all affect that things are fine and to keep fighting. If a tumor completely takes over the B cell population, there's no more friendly interaction with those T cells, and they're missing a relationship and like a growth signal, just to put it small for them. But to say the least, it was a very cool, like it was a single spoken meeting that he showed it at. And it was Dr. Ken Young from Duke, and he just said, we're going to figure more out. And this is exciting. So this was December this year that I saw that, and I was like, that's awesome. It was like one of those moments that I had in here, right? Not to tout Carleton too much, but I'm gonna.
I had a question.
Yep.
I had a more general question. So personalized medicine seems like it's kind of a misnomer because I mean at some point decisions have to be made in the boardrooms at Mayo Clinic. It's like how if it's too personal. Okay, we save one person. How much money can we spend? Where's the cutoff? Or what's sort of
What's gonna happen now?
behind the scenes? How do those discussions happen? I mean, what does it have to be on that graph there to say,
Right.
you know what, we're going after this, we're designing a drug or not?
Yeah. So the risk of entering any personalized trial is going to be you are deviating from the stat population. You're entering our prediction. And the word money was uttered as well. Do you think it's better for the drugs companies? And like, hey, and like I said, I work for the drug companies. We don't have to pile all evil on, but like a little sprinkle of evil to say the least. But is it more lucrative to have a single treatment? I'm going to use this, or 50 things that we all have to manage? One, the consolidation. Now, so a good union between the personalized medicine is eventually going to be versions maybe with a single backbone of a single drug, but modifications to that backbone potentially. That would hopefully be the best union. Because you're right, we can't sequence in TME micro scRNA every single patient. And the true hope, and especially what I want to do for myself, is that I want to be able to use these signals that we find in the genes, but give something very translatable, like one cytokine that we can look for in ELISA, one stain signal that we can just do a pathology stain for and translate that hopefully a little better. Yeah.
It's gonna be tough calls to make for those physicians and drug company executives, et cetera. I mean, it's not the people, right? It's not your, it's the difference between statistics and being individual. To make those-
It's hard. It's definitely been, well, I had some clinical trial experience, like at UNMC, I got to help open one. It was a little more raw than I was ready for, because is it working or not? And eventually you would stop seeing samples from some people. And that was the reality, and that was tough. To begin also on this, and this can build, we may not have, there may be certain populations that we can't really cure. But the hope, and this isn't a perfect world, but in the hope in 10 to 20 years, could somebody go on drug A for 3 years and then resistance builds? But drug B is here now, 4 more years. Resistance, drug C, and you go on and on and on. That's not a perfect world, but can you imagine a world where cancer is chronic? But you don't die from it, maybe. That's the hope with these jumps sometimes.
Well, Keenan's gonna hang around here for
Yeah.
a little while longer. Come on down and ask us some questions if you have particular
Yeah.
questions, but let's thank our speakers.